What genetic mutations drive different types of astrocytoma behavior? Imagine a tiny change in your genes could influence how a brain tumor grows or spreads. That's exactly what happens with astrocytomas, a type of brain tumor that starts from supportive cells called astrocytes. These tumors can grow slowly or become very aggressive, and their behavior is largely driven by specific genetic mutations. Let's explore the key mutations that shape how these tumors behave. One of the most important genetic changes involves the isocitrate dehydrogenase genes, known as IDH genes. Mutations in the IDH1 or IDH2 genes are common in certain astrocytomas. Tumors with these mutations are called IDH mutant astrocytomas. These tend to occur in younger and middle-aged adults. They usually grow more slowly and have a better outlook compared to tumors without these mutations. The presence of an IDH mutation is now a major marker used to classify astrocytomas and distinguish them from other brain tumors like oligodendrogliomas or glioblastomas. Another key genetic change involves the deletion of tumor suppressor genes called CDKN2A and. When these genes are lost or deleted, the tumor tends to become more aggressive. In tumors with IDH mutations, if CDKN2A and CDKN2B are intact, the tumor is often lower grade and less dangerous. But if these genes are deleted, the tumor can progress to higher grades and become more difficult to treat. In tumors that do not have IDH mutations, another mutation called the TERT promoter mutation is common. This mutation increases the activity of an enzyme called telomerase, which helps tumor cells keep dividing by maintaining their telomeres. Tumors with TERT promoter mutations tend to grow faster and behave more aggressively. These mutations are often found in IDH wild-type astrocytomas and glioblastomas, which are the most aggressive form of astrocytoma. Genetic amplification of the epidermal growth factor receptor gene, known as EGFR, is also linked to aggressive tumor behavior. When EGFR is amplified, it promotes rapid tumor growth and invasion into surrounding brain tissue. Additional chromosomal changes, like gaining extra copies of chromosome 7 or losing parts of chromosome 10, are also common in these more aggressive tumors. These genetic alterations help the tumor grow and resist treatment. Mutations in the TP53 gene, which normally helps control cell growth and repair DNA, are frequent in astrocytomas. When TP53 is mutated, the tumor cells lose some of their ability to repair damage or control their growth, making the tumor more likely to become invasive. In children, astrocytomas often have different genetic features. Some pediatric tumors involve mutations in histone genes, such as H3 K27M. These mutations define distinct tumor subtypes that tend to be more aggressive and behave differently from adult tumors. Understanding these genetic mutations is vital for diagnosing and predicting how an astrocytoma might behave. Testing for IDH mutations, TERT promoter status, CDKN2A deletion, and other markers helps doctors decide on the best treatment plan and whether a patient might benefit from targeted therapies. While research is ongoing into treatments that specifically target these genetic changes, the mainstays of treatment remain surgery, radiation, and chemotherapy, tailored to the tumor's grade and molecular profile. In summary, the behavior of astrocytomas depends heavily on their genetic makeup. Tumors with IDH mutations tend to grow more slowly and have a better outlook, while those with TERT promoter mutations and EGFR amplification are more aggressive and have poorer outcomes. Recognizing these genetic features helps doctors better understand tumor biology, guide treatment choices, and ultimately improve patient care. Always remember, each tumor is unique, so personalized medical advice from healthcare professionals is essential for the best outcomes.